Hey, so if you are doing bulk RNA-seq data analysis, you probably have faced this MA plot. So MA plot stands for the visualization of a relationship between log to fold change and mean of normalized count. Let me explain it in more elaborately, but in a very short manner. For example, you had DE-seq pipeline where you used the negative binomial algorithm for the further analysis of your bulk RNA-seq count matrix data. Now you have a contrast where you set up the comparison from the control versus treated condition. That's for example perspective. Now you want to see what have changed from the context of differential expression. Basically means the log to fold change and in correlation of mean of normalized counts. And for that, MA plot is perfection. Now, what does it means on this y-axis log fold change for the information? It's actually log to fold change. If you see somewhere log fold change, it is actually log to fold change. People write it on different way. And mean of normalized counts. So if we look at the y-axis, we see the M captures the log to fold transformation of fold change between experimental condition. So if you set in the treatment condition, what had changed compared to the control in terms of the gene expression, we look at this log to fold change. It's calculated by log to mean of treatment samples divided by mean of control samples. On the contrary, if you want to see, okay, what is mean of normalized count in x-axis, then you take the mean of treatment samples plus mean of control samples, which is wholly divided by 2. Let's generate a MA plot in our studio. So for example, this is our code. And you have run your DE-seq entire pipeline. So you did all the normalization and comparison, you ran the standard DE-seq entire process, did the contrast. Now you have your data set, you want to visualize them. So you write it like this, which is plot MA, and you go to the DE-seq repository, the analysis, and you set the graph parameter. So if you run it like this, you'll get this. DE-seq analysis interpreted the final MA plot. So let me explain this entire figure. So as we talked earlier, the left side is the log to fold change, and the right side is mean of normalized counts. The dots represents your gene. Now This is the middle line. Now, from this line, the more deviated they are, for example, these and these, the higher the log two-fold change, the better differential expression they have. Either they are upregulated in here or they are downregulated in here. So this log two-fold change from 0 to 10 and positive and 0 to negative indicates how differentially expressed, and in terms of log fold change, log to fold change, how far this specific genes are. If you see at that video we have already explained the value from percentage to log to fold change and vice versa. That's if a one-fold change, log to fold change, how much is it is in percentage, it explained in there. Now when you see and look at, okay, this change is highly expressed and it's highly upregulated after the treatment, and in here it's highly downregulated after the treatment compared to the control. Now, what is the importance of the mean normalized counts on the context of log to fold change? The importance of mean of normalized counts is looking at the overall expression of your gene. If the gene is highly upregulated, but their mean of normalized count or their overall expression is very low, that probably have less significance. But if the log to fold change is highly upregulated and their mean of normalized count is quite good, then these genes are really good representing upregulated genes or differentially expressed genes. That's the kind of a summary of this entire MA plot. I try to keep it as short as possible, but if you have any question or concern about the explanation of the figures as well as the code, feel free to ask me, send me email, or just comment it. Thank you.